Now, the second story I want to tell you now in this regard is diabetes. So we've done arthritis, this cascade. We don't know what causes arthritis. We still don't know what that question mark is for arthritis. We think we know what it is for diabetes. So the second story is diabetes. And this is the discovery we made last year. We published this last year, a big paper we had out in this, and it was a great thrill to make this discovery. And diabetes is a bit similar in many ways, inflammation again. And this is especially about a second molecule called IL-1. So TNF is very important, and the drug that's made in Grange Castle blocks that. IL-1 is also very important, and it gets overproduced in inflammation. It's another inflammatory signal. And I'm going to tell you all about IL-1 now, specifically, as if you like, it's another domino in the cascade. And IL-1, I worked on IL-1 in the mid-80s. Many of us did. By 1990, it was out of fashion. People stopped working on it. Do you know why? Because anti-TNF was working. They had a drug. I forget IL-1, you know, and people stopped working on it. 6-7 years ago, a resurgence of interest in IL-1 again, which is wonderful if you'd worked on it in the 80s. And why did that resurgence happen? Well, very interestingly, again, a key tile was found in the profile that drives IL-1 now, and it's called NLRP3, a horrible name, I know. But again, another one of these inflammatory proteins was found that drives IL-1, and that reawakened the field. That discovery, that particular little domino, reawakened the field. And in the case of diabetes especially, you have NLRP3, the tolls again are here, you have IL-1, and then we get, the hallmark of type 2 diabetes is insulin resistance. Your body makes insulin after you've eaten something, and that helps you absorb glucose. If you've got type 2 diabetes, you're insulin resistant. Your body's not responding to insulin. And for some reason, the inflammatory protein, we don't know why, makes you insulin resistant. The beta cells in the pancreas die, and you get type 2 diabetes. And all these tiles end up as type 2 diabetes. And I want to tell you that story, because we found what the question mark is here, which is what I'm telling you. Now, how was this discovered? Well, again, it's a funny story, this, and I know the guys, Hal Hoffman was a key guy in this. The NALB3 gene was discovered, first of all, and it was discovered in families who get horrible multi-body inflammation. It's very rare. There's a series of diseases where the whole body becomes inflamed, and it was inherited from generation to generation. And they mapped that gene, and it turned out to be this awful word in LRP3. And all these diseases, this Muckle Wheless is one of them, have a mutation in that gene. And what that means is that protein, NALB3, is now turned on in those people. There's a mutation. And it's jammed into the on position, driving IL-1. And IL-1 is causing arthritis, it's causing diabetes, it's causing all kinds of things. And that genetic discovery opened up the field. And then it was realized the normal NALB3, its job is to sense, again, noxious substances in the body. It can sense bacteria, it can sense viruses. Very importantly, it senses particles. So for instance, asbestos is up here somewhere. That is sensed by NALB3, and asbestos causes lung inflammation. Silica is sensed by NALB3. Beta amyloid, a very important factor in Alzheimer's disease, that's sensed by NALB3. And the output is IL-1. And again, it's almost as if the body is trying to counter this imbalance by driving NALB3 and driving IL-1. So its discovery was very important for the IL-1 system. And let me give you a really good example of this. This is one of these horrible diseases that were inherited. It's called Muckle Wheless disease. This is a disease where people in their teens begin to get fevers. It starts with that, like the flu. They then begin to feel very sick in their joints. Their kidneys stop working. By their early 20s, they're dead. And there was no treatment for this disease. Hal Hoffman finds the gene. It's NALB3. He figures NALB3 drives IL-1. Give these patients an IL-1 inhibitor. They're cured. Amazing, right? And it's a rash. This is someone, they get a horrible skin rash. They get horrible eye inflammation. A single injection, 24 hours later, all the rash is gone. And the eye inflammation is gone. These are markers of inflammation in the blood. They go to 0, and it lasts like 4 or 5 weeks. Now, that was a life sentence before that discovery. So here we have an example of a really good discovery. Now, it's very rare, this. But one day, all diseases, if we get this level of insight, should be like that. And remember, this is a strange one. This is a mutation in NALB3. Very rare. It's rare because it's so life-threatening, you know. But that discovery gave us this inspiration. Many drug companies start to block IL-1. There's 3 inhibitors in development. They're called cryopyrinopathies, the great name, these horrible inherited diseases. Gout, diabetes, arthritis, Stills, Behçet's, ankylosing. All of those involve NALB3 and IL-1. And it's provoking drug companies to do these trials. Now, interestingly as well, herbal remedies. People often knock them and say it's all nonsense. Feverfew, I worked on that as a PhD student, it's a herbal remedy for arthritis. It's got, again, it's chemistry. It's got a chemical called parthenolide. Guess what? That's anti-inflammatory because it takes out NALB3. So parthenolide is a natural chemical that blocks NALB3, and that's in development now as a possible NALB3 inhibitor. So you never know where these drugs might come from. Now, the last example, the sort of science I'm going to show you now. What would drive NALP3 in type 2 diabetes? Now remember, its job is to sense bacteria or viruses or noxious chemicals in the body. And uric acid is a good example. In gout, if you ever heard of uric acid builds up in your joint, that irritates NALP3 and drives IL-1. And anti-IL-1 is now working in gout. What about diabetes? No infection here. What would be driving NALP3 in that situation? And we turned to this because there was evidence of IL-1. The output from this is indeed diabetes. It was shown 20 years ago IL-1 beta causes insulin resistance. It also kills the beta cells that make the insulin in the first place. So it's very damaging in type 2 diabetes. So we knew this was going to be important, and there have been a couple of trials already blocking IL-1 in diabetes. This is one of them. What they measure here is sugar in your blood, and it's high in the diabetic. You give anti-IL-1, it gets normalised. That's blocking IL-1 in type 2. So we know this was a good thing to go after. The question though is what was driving it, and we turn to this stuff. It turns out that if you take someone with type 2 diabetes, their pancreas is chock full of this. This is called IAPP. It's a protein that gets deposited in the pancreas in type 2 diabetics. And we said that must be the irritant here. That must be irritating NALP3 and driving IL-1. And here's an example of a diabetic pancreas. All this red stuff is this IAPP. It looks a bit like beta amyloid in the brain. It's similar to the protein that causes Alzheimer's. And we said, I bet this is driving NALP3. And lo and behold, that moment on a Friday afternoon, the postdoc, I said to the postdoc about a month before, do this experiment, didn't hear from him. A month later, look at this result. And we get these, it's like scoring a goal in the Aviva Stadium when you get a result. This was what we were looking for. This is IAPP. Driving loads of IL-1. There's the control. Lots of IL-1 comes out of it. These are known activators. And look at this. We had a mouse that lacked IAPP. No effect. And that's a really good piece of evidence that IAPP is driving IL-1 through NALP3. And then even better, we made a mouse model. Now this can be seen as bad if you're an animal rights person, but you've got to use animal models to get these complex diseases. You can make a mouse that overproduces IAPP. So we put the gene for IAPP into a mouse. That mouse gets type 2 diabetes. The IAPP gets deposited. All this red stuff is the pancreas of this mouse, full of IAPP. And look, stained for IL-1, green stain, full of IL-1, and they co-localize very nicely. So we could prove in a model that the IAPP was driving IL-1, which was very nice. And we also showed it was macrophages, this nasty cell that Meshnikov discovered. They go into the pancreas and they try to digest away all the IAPP. And you can stain for macrophages, all these little red dots, you can't see them, the green, the yellow. You can just about see that is macrophages staining up for IL-1. So we knew then that this was the offending agent. And then we published our paper, and then, lo and behold, on Wikipedia, again, it's always nice on Wikipedia mentioning it, we saw this entry, and it says here, if you can read it, it says, it's found that oligomers of IAPP, a protein from amyloid deposited in the pancreas during type 2 diabetes, triggers the NALP3 inflammasome, generates mature IL-1. Right, so in other words, early on, it seems to be a cause for type 2. Now, my mother put that in. No. But this looked as if this was important. And why do we think could be a cause? And this is very good, this bit. We can now explain type 2 diabetes in a stepwise process. It starts with low-grade inflammation. So that's the first step in type 2 diabetes. What causes that? We know this already. It's caused by fat and sugar. Too much fat and sugar irritates the immune system. And this has been known for 30 years. That gets the inflammation going. Okay? And what does inflammation cause? Insulin resistance. So the body stops. Now this is sensible in a way, because if there's loads of fat and sugar in your body, you don't want to take it up into your cells, because that could be damaging. So the insulin stops working, because insulin's job is to allow fats and sugars to be taken up after a meal. So this is why the inflammatory process probably is limiting that, to stop that happening too much. So it's initiated by obesity and by fats and sugars in your diet. The beta cell, to compensate for this, starts to make more insulin. So the beta cell can sense this imbalance. And IAPP turns out to be a hormone made with insulin. It's a naturally occurring hormone, and it helps insulin do its job. So they're made together. The trouble is, IAPP, when it's made at high levels, starts to form these nasty clumps. So it's an accident of nature that this protein begins to clump. It's just its feature, and it starts to clump and you make too much of it. What does that do? That really drives macrophages crazy. They start, NALP3 senses it, IL-1 beta gets made, you get this massive overproduction, and that leads to more insulin resistance, and very importantly, starts to kill the beta cells. So the very cells themselves start to die. And finally, we get type 2 diabetes. So you can imagine in the thrill of this, we have found a key tile in type 2 diabetes. IAPP is nudging over that NALP3 protein, driving IL-1, and then driving all the events down to type 2 diabetes. Now, why is this important? Well, you can imagine. I mean, first of all, this makes drug companies happy because they're going after IL-1 anyway. They knew it was important, but now we have a reason for them to go after it. It would also suggest that an ALK3 inhibitor, a more specific drug than to block that tile going over. The earlier you can go with disease, the better. You'll stop all the tiles falling over. And our company Upsona are developing ALK3 inhibitors to stop this first tile falling over. So really, that discovery may well turn out to be very useful for new therapies for type 2 diabetes. But very importantly, it's not just diabetes. Now remember I said inflammation is everywhere. It turns out NALK3 is in many diseases. It's not just type 2 diabetes. Now the trigger could be different. And here's some examples. In atherosclerosis, cholesterol crystallizes when you've got this atherol. That activates NALK3. Other labs have shown that. Alzheimer's, beta amyloid, is irritating NALK3. AMD is a disease of the eye. Druson's, Huntington's disease, the protein Huntington. Parkinson's has a disease called Parkin. And then motor neuron disease, I think called SOD1. All of these start to get overproduced. Now, why they would get overproduced, we're not sure, of course. I mean, we know in type 2 diabetes it's the fat diet causes this initially, and probably causes the cholesterol bit. Why do you get amyloid in the brain? We don't know. Why do you get SOD1 and Huntington? We don't know. But we know that these are activating NALK3, and they're driving IL-1 production. And the inflammatory process destroys your brain in Alzheimer's, or in Parkinson's, or in motor neuron, your nerves in the motor neuron disease. So this NALK3 process is more than just type 2 diabetes, and again justifies why you want to develop an ALK3 inhibitor. So really, the future prospects for all of this are very interesting, right? And I guess the take-home message overall is that all of this basic research, which is guys working on fruit flies or mashing up tissue from a pancreas or whatever it might be, and using genetics to clone genes and making strange mice that get diabetes, all of that has to give rise to new treatments, hasn't it? Because that's where they'll come from, and stop people putting heated sea sand on your knee. So excellent prospect for new treatments, new vaccines. I haven't gone into that. New, and cancer, very interesting. I mean, I often give cancer as an example as well. There's no doubt now that many of us believe we'll cure cancer through activating the immune system to fight the tumour. So when you have a tumour, it's your immune system that clears it normally, right? And the more we understand about that, the more likely it is we get a cure for cancer through the immune system. Vaccines, very important. There's no vaccines for HIV or malaria or TB yet, so there's lots of work going on there. And then these inflammatory diseases. Many drugs in development. There's about 100 drugs, if you take all the drug companies combined, in phase 3 for all of these diseases, right? So it's a very interesting prospect now. Pfizer, who I do some work for, they have 22 drugs in development for things like lupus, MS, motor neuron disease, rheumatoid, okay? And these drugs are at different stages. So they're all kind of behind this, and they're beginning to see effects with these new drugs. So the prospect really is that we will have treatments, and understanding the causes then will get better, the environmental influence. I mean, we still know what causes them really. So what causes rheumatoid is not clear really. I mean, in the case of diabetes, it seems to be the high-fat diet is the problem here. What causes Alzheimer's? We don't know. But it must be environmental, and there must be some genetic background, like why one person gets it or not. So let's say we both get exposed to some environmental toxin, and I get Alzheimer's and you don't. That's because I've got slightly different genes in my body. And the genes are probably in the immune system that are dysfunctional. They're not working quite so well. And that's enough then for me to get the disease and you not. So the cause ultimately is going to be some environmental influence on the right genetic background. And that really would be the answer as to what causes a disease like rheumatoid arthritis. And those insights then of course feed back into drug development. And that the example of NLRP3 being found in a genetic disease is a good one, because that sparked all this research then into NALK3 and how it works and all the rest of it. So the future prospects I think are very hopeful. And this was in the Daily Express last summer, death rate at all-time low. It's a great headline, isn't it? That's mainly because of statins. Have you heard of those? So statins are used for heart disease, but statins are anti-inflammatory. So these drugs that treat inflammatory diseases are lengthening the lifespan. And the prospect has to be, say, 30, 40, 50 years from now. We're all going to be very old in Europe because the aging profile is going up. Hopefully, as I said earlier, we won't be burdened with these diseases. And that'll be nice, because obviously it's nice to get old, but not if you've got very bad rheumatoid or whatever, or Alzheimer's. I mean, that's terrible. So this research will help prevent these diseases from happening, is what we would hope for. And let me just finish on a couple of things. I often get letters from whenever I give a talk, sometimes you get a letter, or I've been on the radio a couple of times, and someone always writes to you. And it's slightly witty, but it's also sad. And the reason why it's sad is there's a massive level of suffering with these diseases out there. Children with juvenile arthritis, you know. And some of these drugs work a bit, but not as well in some patients. And the second thing that strikes me is the doctors aren't telling the people all about these diseases, you know. And I say, have you not spoken to your GP or your rheumatologist? I couldn't understand a word he was saying, you know. So there's a huge a need for information and to explain to people what these diseases are. And so they're driven to write to people like me, and they say, oh, can you help me? And really, this is the punchline, you know. If we can understand the immune system, we'll find much better treatments for cancer, infectious disease, malaria, RA, MS, colitis, SLE. That's the prize, isn't it? That's why we're in the game in the first place, I suppose. And you have to apply science, very important, to understand the immune system. And that's why science is so key, because we use scientific methods to understand these processes. And that's the real hope. And I'll finish by just thanking the people. This is my lab at the moment. I've got 22 people working down there. You get an idea. It costs me about 1.1 million a year to run my lab, to keep this team going. It's an expensive business. Thankfully, we've got Science Foundation Ireland and Ireland. I've got European Union money. Pfizer would fund a couple of people. These are all postdocs. So all these names here, they've already got their PhDs. These are doing their PhDs with me. And Seth Masters, just to single him out, he did the IAP, found the IAPP discovery. And we must give him, he's an Australian guy. So that's the team of 22 people. And I want to thank them. And very importantly as well, it's a very collaborative. The paper we published on this last November, I think it was about a year ago, there were like 8 different countries on that paper, our collaborators. It's a very international. Like you go to the guy with the mouse, the IAPP, he was in Seattle. I got the mice off him. It was a Japanese lab, had a chemical that we got. And these are all the labs. I mean, there was one in Washington, Kyoto, UCD was with Phil Newsom, Michigan. You know, so it's a very international game. And SFI are the key funders. I always thank them for providing us with the money. But let me finish with one last quote from the book. This is my favourite. Remember the book with the dodgy answers? This is my favourite one. There were 300 students in year 10. Mary and Mark want to find out year 10's favourite colour. Mary asked 30 people. Mark asked 150 people. Mark says, my conclusions are more likely to be reliable than Mary's. Why does Mark think he's right? And someone wrote down, because Mark is a man. And that's the best reason of all. And thank you very much for your attention.